Hey everyone, welcome back to my YouTube channel. If you're a new subscriber here, welcome and thank you for tuning in. If you're an old subby, welcome back. In today's video, I'm going to be talking about what I do. A kind of generalized question that I always give people when they ask what I do. I say that I do HIV research, but that's such a broad and vague answer. So it's so funny because a friend of mine, she always says, I really don't know what you actually do. Like when people ask me what you do, I always say, usebenza ka HIV, which means like that you work ka HIV, you work in an HIV organization. And yes, I do HIV research. So in this video, I'm going to be delving into exactly what does it mean and what do I mean when I say that I do HIV research. So if you're interested in this video, keep watching. Okay. So as I said in this video, I'm going to be delving into what I do. Like I said before, a generalized question that I always give people when they ask what I do, I say, I'm a research scientist. I'm doing research in HIV. I'm really going to be unpacking what that means. So to those of you who don't know, who haven't watched my previous videos, like getting to know me, I am doing my Doctor of Philosophy in Medicine with particular focus on virology and specifically HIV, which is where the answer comes from. What do? I do HIV research. I'm sure you guys know HIV stands for a human immunodeficiency virus. In Africa and in sub-Saharan Africa particularly, we are gravely affected by HIV, and in sub-Saharan Africa alone, we account for 70% of the global infections worldwide. I think globally there are just shy of 37 million people who are infected with HIV. We're less than 1 million new infections now and about less than 800,000 HIV AIDS related deaths. So HIV is still a major problem in sub-Saharan Africa. So what I do as my project focuses particularly on HIV research is I'm particularly focused on HIV cure. So generally in HIV research, people are either looking for a vaccine. So a vaccine is for people who are uninfected, how they can get vaccinated and not get HIV, and a cure. So that is totally eradicating the virus in already infected people so that they become cured from the disease. So in cure, it's further divided. So there is a functional cure and an eradicating cure. So a functional cure would be that we discover a cure that would allow people to stop treatment. As you know, people who are infected with HIV go on antiretroviral therapy, and this is a cocktail of drugs that slows down the replication of actively replicating virus in their bodies. People who are on ART and are taking it well and are adhering to ART can have viral loads that are very low, that are below the limit of detection, and that reduces their chances of them passing it on. So that reduces the chances of transmission as well. So in a functional cure, we want to allow people to be cured of HIV, but they still they still have virus in their body, so they're not 100% cured. So a functional cure would be eradicating ART, so that they don't have to take ART for the rest of their lives. That's a functional cure. And then an eradicating cure would be totally getting rid of all the virus in an infected person's body. That would be an eradicating cure. This kind of cure is going to be particularly difficult to achieve because when a person becomes infected with HIV, we have cells that are latently infected. So what this means is that these cells are infected with HIV, so the HIV has been able to enter the cell and take over the genomic machinery of the cell, but they're not producing viral protein. So this cell is not producing new virus. However, the cell remains infected. That is a major barrier to cure that we're facing today, is the presence of latently infected cells, and these latently infected cells reside in an HIV reservoir. So reservoir eradication is a major barrier to us achieving an HIV cure. Do I believe that we will get an HIV cure in my lifetime? Absolutely. I absolutely believe that we will be able to get a cure, whether it's a functional cure or an eradicating cure. Absolutely, in my lifetime. I am optimistic, and I'm 100% going to say that yes, in my lifetime we're going to see an HIV cure. Yeah, so my research is more focused on HIV cure. So I'm looking at a particular gene in HIV, which is called TAT, TAT, or the trans activator of transcription. So usually during HIV replication, this gene plays a major role in enhancing viral gene transcription. So it plays a major role in making sure that new variants are produced at a particular rate, in layman's terms. So I'm looking at this gene and looking at how genetic variation within this gene, so how the genomic sequence of the TAT gene changes, and how this has an effect on its functional activity. So for example, if a gene has a mutation at position 4 of an isoleucine instead of a valine, these are amino acids that I'm talking about, does this affect its ability to transactivate the HIV promoter? So does it affect its ability to make transcription happen at a slower or a faster rate? In layman's term, that's what I'm looking at. So that's what I've looked at in my master's project, and now in my PhD project, I'm going a little bit deeper and digging a little bit deeper into this whole phenomenon. I will try and say it in layman, in simple terms, so you guys understand. So basically, I spoke about the TAT gene. So what happens in HIV replication is that the TAT gene it binds to the HIV promoter. So it binds to this element that is called the transactivator response element. It binds to that element, and when it binds, it recruits. So it calls its friends, its transcriptional factors, and it's like, hey guys, come along. It's time for HIV replication to happen now. So it recruits transcription factors. So these factors play a role in moving along HIV replication. So particularly in these factors that it recruits, now I'm looking at the PTFB, or the positive transcription elongation factor B. This plays a major role because it is actually a component of this that hyperphosphorylates the RNA polymerase, which is the enzyme that is used to carry along mRNA elongation. So I'm looking particularly at that and at TAT genetic variation. So how do these two work synergistically, and how do variations in TAT affect TAT's ability to recruit the friends? So remember the friends that I was talking about, when it binds, it recruits the transcriptional factors. So how does the different variation in TAT affect its ability to call its friends, which is the transcriptional factors during HIV replication? So that's what I'm looking at now in particular. That's a very broad overview. There are a lot of different things that I'm going to be doing and looking at, like site-directed mutagenesis. I'm going to be looking at the protein dynamics, protein DNA interaction, protein interactions, protein foldings. I'm going to be looking at different TAT variants in JLAT, so latently infected cells, and do different variations in TAT affect its ability, for a cell to either be actively replicating, so that's producing new virus, and in active replication you have ART, which is able to target these cells, because that's what ART does. Or does it result in these cells being latently infected? So that's when they go into the latent viral reservoir. That's another component of my project that I'm going to be doing, looking at latency and its propensity for latency as well. So I hope that explains generally what I do right now when I'm doing HIV research. So that involves me actually being in the lab. I have previously amplified TAT from acutely infected, so HIV acutely infected patients. We work with HIV in the lab that I work at. We extract RNA from the plasma, which is a component of blood. We extract RNA from the plasma for all our downstream applications. So I am totally hands-on in actually when I say that I work with HIV, I really do. And that everything that I've explained is kind of what I mean when I say I'm doing HIV research. So I hope that people now, especially my friends, if you're watching, they understand now what it is exactly what I do. Previously, I have done my honors in microbiology, so that was like the study of bacteria, and then I also did genetics in my undergrad, so I have a bit of that background. And I also currently do genetics, so that's in the TAC genetic variation and stuff that I spoke to you guys about earlier. So I'm a geneticist, a microbiologist, and a virologist. So that's what I'm doing right now, is the study of viruses, and in particular HIV. So that's what I'm doing right now, and I hope it was clear, and I hope that you guys learned something new about HIV in this video. And I just wanted to touch a little bit on, you know, what's currently happening in terms of HIV in the news. I'm sure you guys have heard of the patient that now is the second patient to be supposedly cured of the virus, and I just wanted to explain to you guys what that actually means. This is not like a huge scale kind of way that we can apply to everybody to cure HIV. So basically what happened with this patient, which was the first patient, was the Berlin patient, which is similarly what happened to this patient, the London patient, is that this patient, there's a very rare mutation. Less than 1% of the population has it, and that is a homozygous deletion in their genome that doesn't allow the HIV to bind to a receptor called CCR5. So during HIV replication, the virus needs to bind. So in the cells in the body that it infects, there are receptors on the cells. So think of it as like Lego pieces, I guess. So there are receptors, so the little HIV virion, and the cell, you know, has receptors on it, and then the HIV comes and it binds to the receptor on the surface of the cell. So basically this person that has been supposedly cured of HIV doesn't have this receptor that the virion would come and bind to. So when the HIV is floating around in the blood, it doesn't have anything to bind in, so it can't infect the cells. And that's a major mechanism by which HIV works in destroying the body and destroying cells in the body, as well as producing new virions. It needs to bind to the cell, enter the cell, inject its genomic material into the cell, and take over the genomic machinery of the cell. So it now instructs the cell, instead of it being before a healthy cell that was producing healthy proteins for the body, the HIV comes on, it binds, injects its genomic material into the cell, and takes over the genomic machinery. So now the HIV is telling the cells to produce a new HIV protein, so little virions that will be produced and formed within the cell that it's infected, and then bud off and then form new HIV virions that will go and infect new healthy cells. So that's how HIV works. So this person that doesn't have the receptor, which means the HIV, it just floats around, it cannot infect the cells in that person. So that person then does not become sick. And the way that they, you know, got this is through a bone marrow transplant of a donor that had this mutation, and then because these are stem cells, they are injected into this person. These were cancer patients, and then these new cells are now the cells that take over the body. So now these new cells are mutant cells without the receptor that I spoke about, and thus HIV cannot bind to these cells. So that is how we've had these cases where people have been cured of HIV. And I hope that you guys can realize that this is not something that we can apply on a large scale. We can't just give everybody infected with HIV bone marrow transplants. That would just be absolutely crazy. It's a major operation. Already there's shortages of bone marrow transplant donors, so it's just not something that we'd be able to apply on a large scale. So I just wanted to clarify that and tell you guys what exactly happened in those studies and in those people that have been supposedly cured of HIV. I will be doing a more basic HIV 101 video in the future, because I feel like there are a lot of myths and there's a lot of things that young people don't know that need to be debunked. So I will be doing a video like that in the future, but I just wanted to do this video and let you guys know what exactly it is I do and what I am doing when I say I'm doing HIV research. So I hope you enjoyed the video, and thank you again for tuning in. If you're not already subscribed to my channel, please go ahead and click the subscribe button and the notification bell so you can be notified when I upload new videos. Thank you so much again for tuning in, you guys, and please leave me a comment down below. If you have any questions for me or comments, please comment down below and engage with me on all my socials, which are linked down below. I will see you guys on the next video.